T minus 6, 5, 4, 3, 2, 1.
On December 5th, 2020, the SpaceX Dragon cargo spacecraft is scheduled to launch on a Falcon 9 rocket from NASA's Kennedy Space Center in Florida. It will be delivering science investigations, supplies, and equipment to the International Space Station. One of the deliveries will be a new technology developed here at Louisiana Tech University by Dr. Gergana Nestorova, an assistant professor of biology in the School of Biological Sciences. While NASA is taking steps to expand space exploration, they need to ensure astronauts on longer missions, like to Mars or to the Moon, will have all the resources they need to successfully complete their mission. One of those resources is food. Fresh, nutritious food that will help sustain the astronauts in an environment that will not. Right now, astronauts are dependent on regular shipments of freeze-dried and pre-packaged meals. But the farther they travel from Earth, the longer it will take for them to receive those shipments. These meals, like other food, will break down and over time become less nutritious. If astronauts have to survive without a new shipment for months or years, their prepackaged meals may spoil before the next shipment's arrival.
So we are trying to develop a product for astronauts to use on the International Space Station for gene analysis. Right now, their gene analysis system is very complicated, and it involves dissecting biological samples, mixing chemicals, and testing the RNA that way. Our product is a functionalized needle that you basically just poke into a biological sample, like a plant matter, say a radish, which is what we're using right now, and test it directly from the needle. Through the development of our product, we have to test its efficiency. So it's not so much about sequencing the DNA of the radish as it's more about how much can we get our needle to pick up. So that's what we're testing when we repeat it over and over again.
So it's more about making sure that you have an accurate reading?
Yeah, exactly.
Okay.
So our whole project is to perfect this product so that it can be used on the International Space Station permanently, or until something better comes along.
So the goal of this project is to test the feasibility and the compatibility of this single-step extraction method for purification of genetic material. So we want to test whether this method is compatible with the instruments on ISS. So to do that, we had to modify the attachment technology to increase the long-term stability of this probe so they can be stored for long periods of time on ISS. And so the main goal is to test whether this method is compatible with the instruments, and if that is compatible, if everything works well, then this could become one of the tools that is used on ISS for purification of genetic material.
Hopefully, the technology Dr. Nestorova developed will allow NASA to more efficiently observe and analyze the plants grown on the ISS, so that space farming can progress and astronauts can build a sustainable habitat for themselves on the Moon and eventually on Mars.